Hello everyone, I'm very glad to participate in this conference. The topic I'm reporting today is BENviewer, a gene interaction network visualization server based on graph embedding model. My report will be divided into the following 5 parts: introduction, method, evaluation, result, summary, and application. The next section is introduction. As a powerful biological knowledge discovery tool, network information visualization can assist researchers in discovering the biological explanations of interesting gene set, or even the entire interaction network in an intuitive way. However, the present visualization tools such as Cytoscape can only show the topographic structure of networks. It cannot visualize the information behind each node and edge in the network. Proteins or genes with interaction or participate in the same biological process should be close to each other and form a cluster together in a visual space. However, the present method do not utilize the knowledge of this property. Graph can be defined as a set of nodes and edges between nodes. A graph embedding model can mapping the node and edge attributes in the network to the low-dimensional space for visualization, which has been widely used in natural language processing. Embedding the above knowledge in a 2D space and show it in a graph would greatly facilitate researchers to explore the biological meaning of genes. In this study, we developed a novel visualization tool which could facilitate research into the biological meaning of a given gene list. For example, when a gene list is obtained by differential expression analysis, user can observe the distribution of these genes and whether those genes in the function cluster. Next section is method. We employ 3 embedding methods on reactome pathway interaction database. The reactome database supplies an edge list for all gene interactions in whole human pathways. The graph embedding algorithm including deep work, line, and node-to-vector. Different methods focus on different properties of network. For example, deep work focuses on the direct interaction between nodes. Node-to-vector focuses on the structure similarity between nodes, which means they have a similar local connection pattern. The next section is evaluation. After the graph embedding, testing was applied to reduce the dimension for visualization. 2 evaluation indicators were used to assess the embedding performance from different perspectives. Label-free cluster score survey whether the distance within the cluster of 2 nodes is less than the distance of 2 nodes between clusters. Interaction-based score evaluates whether the nodes pair which has interaction are allocated to the same cluster. Next section is result. For representative pathway, DNA repair, toll-like receptor cascades, complement cascades. Metabolism of nucleotides was used to compare with embedding method and cytoscape force-directed layout algorithms. Figures show that the graph embedding algorithm is significantly better. It seems obvious in figure that by our method, majority of genes involved in DNA repair pathway are clustered together. There is a significant enriched cluster of genes related to the complement cascade pathway, indicating the relative tight and independent interaction between them. Comparison between embedding method and cytoscape force-directed algorithm. The graph embedding algorithm score is better than the force-directed algorithm, both in cluster-based and interaction-based score. It is proposed that our method has the advantage of capture the interaction property of genes. Summary and application. This paper compares the application of 3 graph embedding algorithms: deep work, line, node-to-vector, in realizing the interaction information of biological networks. We also developed a web-based application, BENviewer. BENviewer is a brand new online gene interaction network visualization server based on graph embedding model. With mature graph embedding algorithms applied on several interaction network databases, it provides a human-friendly 2D visualization based on more than 2,000 biological pathways, and those results present not only genes involved, but also tightly of interaction in a intuitive way. Thank you very much for your attention.